Cloning, biology only. Cloning is the process of producing genetically identical copies of organisms, cells, or genes. These can occur naturally, for example, identical twins, or be produced artificially using biotechnology. 1. Tissue culture, small groups of cells taken from part of a plant, for example, leaf or root, are grown on nutrient agar with hormones. Grows into many genetically identical new plants. Useful for preserving rare plant species. Producing large numbers of plants quickly in commercial nurseries. 2. Cuttings, simple method used by gardeners for many years. A part of a plant, for example, stem, is cut off and planted. It grows into a new identical plant. Cheap, easy, and reliable way to produce clones. Cloning in animals. 3. Embryo transplants. Sperm is used to fertilize an egg in a lab. The early embryo, before cells specialize, is split into several cells. Each cell is transplanted into a different surrogate mother. All offspring are genetically identical. Used in livestock breeding to produce animals with desirable traits. 4. Adult cell cloning. This is a more advanced technique to create an exact copy of an adult organism. Steps: the nucleus is removed from an unfertilized egg cell. The nucleus from an adult body cell, for example, skin cell, is inserted into the empty egg. An electric shock stimulates the egg to start dividing, like a normal embryo. The embryo develops into a ball of cells. The embryo is then implanted into a surrogate mother's uterus. The result is a clone of the original adult from which the nucleus came. Famous example, Dolly the sheep, 1996, the first mammal cloned from an adult body cell. Benefits of cloning: helps preserve endangered species. Produces animals with desirable traits, for example, high milk yield. Can produce genetically identical organs for medical use. Tissue culture helps create disease-free plants. Risks and ethical concerns: reduces genetic variation, vulnerable to disease. Unnatural process, some see it as playing God. Cloned animals may suffer from health problems or shorter lifespans. Risk of abuse in human cloning, strong legal and ethical opposition. High failure rate, many embryos die or develop abnormally. Example 6 question, describe the process of adult cell cloning and give one benefit and one ethical concern about it. Model answer: in adult cell cloning, the nucleus of an unfertilized egg is removed. An adult cell's nucleus is then inserted. An electric shock stimulates it to divide, creating an embryo. This embryo is implanted into a surrogate mother. Cloning can produce animals with desirable traits but raises ethical concerns. It may reduce genetic diversity and cause animal suffering.